Hello, everyone. My name is Marek Jutel, and I'm the president of the European Academy of Allergy and Clinical Immunology. And it is with great pleasure that we, EAACI, are part of this fantastic event, and we can deliver our symposium, novel tools developed by EAACI. So we will describe what we are currently doing in the area. APAACI is our sister society, so it's part of our program, and that's why we can be present here, and we also welcome APAACI to our congresses, next one in Prague next year. So we are going to have three speakers, Professor Stefano Del Giacco, who is president-elect of EAACI, Professor Joanna Agace, who is past president, and myself. I will deliver the last talk of the symposium. So the first contribution is from Stefano Del Giacco from Calgary, Sardinia, Italy, who will talk about protocols for data sharing. And then the next talk by Joanna Agace, she will present our work on biological guidelines. As you know, we published and we are still in the process of publishing a number of guidelines on biologics, and Joanna will summarize this work.
Well, good afternoon, ladies and gentlemen. It's truly a great pleasure to be here today. I would like to thank the organizers for the invitation, and I'm particularly happy that we can have again this APAACI joint symposium, which is the fruit of a long-standing collaboration with the two societies, which I hope will last for many years. In the next 20 minutes or so, I will try to discuss about what happened with asthma management during the tough times of COVID-19 pandemics and what the European Academy of Allergy and Clinical Immunology recommended at this regard. This is my disclosure. Well, let's talk about the first wave. Of course, some concerns were raised about asthma as a potential risk factor associated with worse outcomes in patients affected by SARS-CoV-2 infection. The traditional paradigm for which viral infection is the key for asthma exacerbation was still in place, and so there were some concerns about asthma as a risk factor, but also about asthma treatment as a potential additional risk factor favoring the viral infection because of the use of corticosteroids. And so there were also concerns about the possibility of asthma management in the clinic. Immediately, GINA reacted and there was the recommendation of continuing using inhaled asthma controller medication, including inhaled corticosteroids, and patients with severe asthma were recommended to keep on using their oral corticosteroid, but in the lowest possible dose, and when possible to switch them into a biologic treatment in order to limit the need for oral corticosteroids as much as possible. However, we saw subsequently that oral corticosteroids and corticosteroids in general have been beneficial in the treatment strategies of SARS-CoV-2 infection. But let's have a look at what happened during the first wave. These are the data, some American data about what happened among hospitalized patients with severe COVID-19 comparing asthmatics and non-asthmatics. And comparing with a systematic review, this is a nice paper from CTEC and collaborators from this year. They were taking into consideration 389 studies, selecting only 16 with an average age of 40, which can maybe represent a bias. And these are the pool odds ratio of deaths among the patients with asthma compared with those with no asthma. The vertical line indicates no difference, and the odds ratios were favorable in almost all the studies, as you can see from the diamond. For asthmatic patients, only in 2 studies, they were not favorable to asthmatic patients. Again, underlining, however, that the risk of death for asthma patients is even less than non-asthmatic patients. And let's have a look about what happened with children. This is a very nice study from Nikos Papadopoulos, also published in a recent issue of Allergy. This is related to the peer cohort, which is a multinational longitudinal childhood cohort composed by more than 1,000 asthmatic children and more than 500 non-asthmatic children. They were comparing the main asthmatic outcomes between 2019 in a darker colors, blue, darker red, and the lighter blue, lighter red for the pandemic times. As you can see for both categories, all the outcomes were much reduced, so they had much less upper respiratory tract infections, much less lower respiratory tract infections, febrile episodes, emergency visits, and hospital admission. But the notable data are that while in the previous times, asthmatic patients had a higher rate of upper respiratory tract infection, lower respiratory tract infection, emergency visits, and hospital admission, instead, during the pandemic, children with asthma are not disproportionately affected by COVID-19. And let's have a look about the rhinovirus testing data. These are data from the Philadelphia Children's Hospital, and these are the rhinovirus testing data from January 17 to March 17 and March 18 to May 17, comparing the 2 time spans. The first one from 2015 to 2019, so historical data somehow, versus the current year, 2020, the year of the pandemics. In darker blue, we can see how the rhinovirus testing was dramatically dropping after the safety measures were in place, like lockdown, masks, and so on, for the pandemic times, while it was raising as usual in the non-pandemic times. And in these 2 graphs, it's clear that probably the measures were effective in reducing viral infections, including the SARS-CoV-2 infections. And if you want to compare adults data, this is a study on a small cohort of patients from the Netherlands, from the group of my friend Gert-Jan Braunstahl, in which they were also comparing previous time spans like 2017, 2018, 2019 with 2020. And it's a matter of fact that asthma exacerbation prevalence went down during the lockdown. And this represents the mean number of exacerbation per patients per quarter per year. And we have in Q2 a statistically significant difference from Q2 to the other years. So this is clearly a sign that, again, also in adults, maybe a better compliance to treatment together with isolation, masks, and other hygiene measures were probably effective in protecting asthmatics from exacerbations. But now let's talk about immunomodulation and asthma in the COVID era, meaning what EAACI was analyzing of these patients, in particular the use of biologic treatments. So we have to consider that there are 2 main pathways in the pathogenesis of airway inflammation in type 2 high asthma. Via allergen-presenting cells, the dendritic cells present the antigen that expand allergen-specific T helper cells, the Th2, or through epithelial cytokines via the activation of ILC2, the innate lymphoid cells. Together, they represent what is called actually the T2 inflammation. Both cell types are regulated by the transcription factor GATA3, and both pathways can anyway be activated and triggered by epithelial cytokines like TSLP, IL-25, IL-33, which will be future targets for treatment. And anyway, they result in a release of type 2 cytokines like IL-4, IL-13, and IL-5, and enhanced production of IgE by B cells. the immunomodulatory intervention that we have available today are somehow listed to the right in green. We can consider allergen immunotherapy as an immunomodulatory treatment, of course, anti-IgE with omalizumab and very soon ligelizumab, anti-IL-5 or anti-IL-5 receptor, anti-IL-4/13. We can somehow consider leukotriene antagonists probably as immune modulators, and then we will have new tools upcoming, new targets and new drugs to block upstream the paths that can activate this cascade. The question that raised, we probably know the answer nowadays, but is immune modulation with biologics in the COVID era safe? How the biologics can interact with COVID pathogenesis, and what EAACI recommended. So we have several medical societies recommendations during the COVID-19 pandemics. All of them, they were not recommending to stop biologics. They were just recommending a kind of home administration of the biologic drug, but not stopping it unless the patient was affected by COVID. But how T2 inflammation and COVID interact? Let's try to focus on T2 response. T2 response is modulatory and try to modulate the inflammatory response. Is usually unrelated to viral control, but it's surprisingly triggered by SARS-CoV-2 infection. In fact, in patients with COVID, increased serum levels of IL-4, 13, and 5, so T2 cytokines have been described. And making ourselves a question, we can ask if type 2 response can be probably an attempt of the immune system to modulate the inflammatory state. And honestly, I find this graph taken from an EAACI position paper assigned by Alessandra Voltaggio as a first name very interesting. This is the hypothesis of favorable evolution of SARS-CoV-2 infection in the context of type 2 cytokine and regulatory cytokine responses. We can see here the hypothetical evolution of the antiviral immune response during SARS-CoV-2 infection. So the lung injury is propagated by the innate and adaptive immune system with type 1 inflammation here. You can see this is upregulated in the acute phase together with the adaptive immune response, which is activated shortly after the innate immune response. However, the dynamic balance starts between type 2 regulatory and adaptive immune response with the different cytokines, and so the type 2 is prevalent, and the type Th1, Th17 together with the innate immune response are lowered. And this underlines how both high levels of certain type 2 are like IL-4 and IL-13 and regulatory cytokines like IL-10, because eosinophils absolute number and T2 gene signature are inversely correlated with ACE2 expression. So the higher the eosinophils, the lower the expression of ACE2. So it's clear that if we use an anti-eosinophil drug, an anti-eosinophil biologic, lowering the eosinophils, probably we will have a higher ACE2 expression. And this is not only demonstrated on the peripheral blood, but it's also demonstrated through the mean T1 gene expression, which is T1 and T2 gene expression. So the normalized ACE2 expression is much lower in the T2 gene expression signature. Again, so an evidence that both eosinophilia and T2 signature are inversely correlated with the ability of the SARS-CoV-2 to infect patients. And now let's switch a little bit to the real life. This study performed in Israel was showing the COVID-19 risk and outcomes in adult asthmatic patients treated with biologics or systemic corticosteroids. They took into consideration the nationwide database. They were taking out 80,602 asthmatics, of which 514 were on biologics, and they were all tested with PCR for SARS-CoV-2, and out of these, almost 10% of the patients were positive. Well, the evidence was showing that patients previously treated with systemic corticosteroids had an increased risk of worse outcome of COVID-19 and also higher risk of mortality after 90 days from the negative swab, while patients on biologics did not have this outcome and they were overlapping with the ones of non-asthmatic patients. Also here, one can see easily the adjusted cumulative incidence for biologics and steroid use. The higher the usage of steroids, the higher the risk for these patients. So in summary, in the COVID era, the type of inflammation in asthma counts. Typical characteristics of severe asthma, which belong to the most of the patients, are T2 inflammation with its hallmark cytokines IL-4, IL-5, IL-13, and eosinophilia. However, the evidence shown has demonstrated that IL-13 seems to downregulate its2 expression. Eosinophilia is negatively associated with the COVID-19 susceptibility, and eosinopenia is a marker of potentially severe COVID-19. In addition, there is also a role for mast cells, which have a high antiviral potential through interferons and other antiviral mediators. But which are the recommendations from the European Academy of Allergy and Clinical Immunology for patients with allergic disease and under biologicals during the COVID-19 times? If no infection, absolutely maintain biological treatment. If possible, self-application of the drug, and if restrictions occur, follow up via telemedicine with a specialist. If COVID-19 with a true infection occur, biological drug must be withheld, and asymptomatic mild moderate SARS-CoV-2 positive patients with home isolation can continue their background asthma medications, while the severe ones in hospital admission have to prioritize the acute management of respiratory distress syndrome. However, after recovery and once the PCR swab is negative, one has to immediately reestablish the pre-infection treatment after reassessment the situation, because of course if some respiratory problem occur to the patients, maybe the treatment must be revised. And we go to the take-home messages of my presentation. Asthmatics are not at a higher risk of worse outcomes when affected by COVID-19. The use of biologics is safe. They have to continue, but they have to stop if they get infected by SARS-CoV-2. The pathophysiological bases are sufficiently solid to confirm this choice, and T2 high-profile patients seems to be at a lower risk of developing severe forms, while T2 low patients may be at increased risk, so increase vigilance upon developing symptoms. And with this, I thank you very much.
In relation to my talk, I have nothing to disclose. So, when we are talking about biologicals for allergic diseases, we are fortunate because we have now a better understanding of the mechanisms. And this enabled a stratified approach for the management of, for example, severe asthma, supporting the use of targeted treatment with biologicals. However, when we started working on the APAACI guidelines, we saw that many aspects require further clarifications. First of all, the selection of a certain biological, and I will use the example of severe asthma because this is the most complicated choice that we might have, because we have 5 products registered with 2 more in the pipeline, and they all target overlapping disease phenotypes. How we define response? What are the strategies to enhance the responder rate? What is the duration of the treatment and its regimen in clinic or home-based, and its cost effectiveness? So, the current challenges come from the fact that we are using in our research what is called the investigator hypothesis-driven approach, which is the supervised model. That lead to a drug-centered development strategy. Many new treatments developed solely based on their mode of action, leading to what we have many me-too drugs. So, at least 3 monoclonals that are used in asthma target the IL-5 pathway, and none was developed for non-T2 asthma. The third problem that we have is that the patient-centered questions are addressed in a non-systematic and voluntary manner in the post-approval stage by non-commercial entities. Last, and this leads to a serious implementation gap of new drugs in real life. So what is happening? Why we need to step ahead and go from randomized controlled trials into real life. This is the example of targeted interventions in T2 asthma. As you can see, all the monoclonals and small molecules that are currently under development, with targeting different pathways, they all do the same thing. So they have the same effects and they all have the same predictive biomarker. So it's very difficult to choose between them when we have severe asthma patients, for example, with a eosinophilic inflammation. In addition, all the studies excluded, for example, the overweight and patients with several other comorbidities, and this is the reality. 85 up to 85% do not correspond to randomized controlled trial profile. They were patients with low or very low adherence for the background treatment. And there is another problem. There is a higher likelihood of biological use in relation with a higher income, commercial insurance, and access to a specialist. We could have, for example, a pragmatic approach that is currently being used now. This is the perspective of a clinician, but of course it's expert opinion and not evidence-based. So, for example, you can look at the association between different comorbidities. So if you have, for example, the association between asthma, chronic rhinosinusitis in relation with a high eosinophilia, either reflected by eosinophilic esophagitis or hypereosinophilic syndrome, then you might think that the IL-5 receptor alpha should be targeted and use the approaches that are there. If you have the association between asthma, atopic derm, and chronic rhinosinusitis, then you can have a look at the only monoclonal that is currently approved for all three of them. And you can target the IL-4 pathway. Or if you have the association between asthma, chronic urticaria, or asthma with viral exacerbations, where we know that the IgE pathway is important, then you might target this pathway. And currently under development, if you have a non-T2 profile, you can look at the two currently monoclonals that are being developed, targeting IL-33 and its receptor, ST2 and TSLP. So with this in mind, we started developing the EAACI guidelines. I will share with you the process of developing these guidelines and some of the important features of this. First of all, like for every other guideline, we use the grade approach, but we separated the recommendations for each biological and for each outcome. So you will see that, for example, for exacerbation, you will have one recommendation, for lung function, another one with different strength of evidence. Then we try to propose a management algorithm for all the diseases that we included for atopic dermatitis, for chronic urticaria, for CRS, and for severe asthma. And of course, highlighting future approaches and research priorities. I will give you an example so from now on I will use the example of severe asthma, because we have to keep up with the time. So the structure questions for the systematic reviews here were looking at all three asthma phenotypes. So we look at severe eosinophilic asthma, severe allergic asthma, and severe T2 asthma. So all the three asthma phenotypes that benefit currently from a biological. And as per grade methodology, you have to stratify the importance of the outcomes by scoring them into critical, important, or low importance. And we choose as critical outcomes exacerbations, asthma control, quality of life and safety. Important, lung function, because if it's not a bronchodilator, FEV1 is not a primary outcome. Decrease in ICS and the OCS dose and rescue medication use. And of low importance, the biomarkers, because they are not very well validated, like FeNO, sputum, and blood eosinophilia. We also had a lot of clinical questions which were not covered by the systematic review. For example, what happens with the dose or route which is not approved by regulatory agency, or if the biological is not approved, for example, the ones targeting the epithelial-derived cytokines. What is the relevance of real-world patients for the clinical trial results? As I have shown you, there is a big discrepancy. Several issues related to the efficacy and safety in the pediatric population, long-term safety, immunogenicity, and in relation to that, the best method to monitor anti-drug antibodies. The joint treatment of comorbidities, as I have shown you in the practical approach, usually you should think in terms of several comorbidities at the same time. The use of biomarkers in guiding the treatment or in the selection of the patient. What are the continuation and discontinuation criteria? What are the switching rules if the biological is not working? Defining efficacy, time to achieve efficacy, how long you have to wait, how long you have to treat, combinations between biologicals, effects of after administration in the emergency department, and health economics, data which were not included in the systematic review. I'm sure that you are familiar with the grade recommendations, what they mean. So for example, for patients, for clinicians, and for policymakers, if you formulate a strong recommendation or a conditional recommendation. So just as an example for clinicians, because we are here today with the clinicians in the room, a strong recommendation means that adherence to this could be used as a quality criteria or a performance indicator. If it is a conditional recommendation, different choices will be appropriate and they will be personalized to each patient. And decision aids may be helpful. Especially, you will see the decision making, the shared decision making with the patient. I will give you one example of how recommendations are formulated for atopic derm, chronic urticaria, CRS. It's the same approach. So we said per biological and per outcome. And you see here each of the asthma outcomes separately mentioned with different types of recommendations. So for example, you can have a strong recommendation for reducing exacerbations and a conditional recommendation for lung function. Two more important features here. You can see an asterisk. So we define the population that is covered by these recommendations as very tightly, using the definition that was used in the clinical trials that were used to do the systematic review and get the evidence. And another asterisk where we say that although, for example, in improving lung function, the effect size is small, and this is why we have a conditional recommendation, this might be very important for patients with very low lung function. I was mentioning clinical decision algorithm because we think that for the implementation of the guidelines, the clinician should have the guidance. So we looked at the triple pillar decision based on phenotypic traits, biomarkers, and outcomes. To that, you add the shared decision making with the patient in presetting the level of achievement that you would like to reach. For example, 50% reduction in asthma exacerbation, or 100% improvement in lung function, or let's say zero rescue medication use. And you start a biological based on all four these areas. Then you assess after 4-6 months. This is an arbitrary cutoff. Maybe for some patients it's not too long enough, but it's an expensive treatment, so you have to put the cost on the top. If the patient is a responder, again, the responder is defined as the preset target for each together with the patient. Then you continue the treatment, you monitor safety and collect data for further from real life, what you are doing and treating. Ideally in a registry, looking at some good criteria like the relevant criteria and preparing for the update of the guidelines with what you collect as good quality data from real life. If the response is suboptimal, then you have to go in at the level of the airways and look if there is persistent eosinophilic inflammation and airway hyperactivity. If there is neutrophilic inflammation or no inflammation, it's clear that you cannot continue targeting the eosinophils and you have to stop the biological and treat like COPD with triple therapy or in selected cases bronchial thermoplasty. If there is persistent eosinophilic inflammation, you have several possibilities. First is check the adherence, because usually the patient forgets to use the background controller treatment and fully believes that only one injection with a biological is enough. And this is why the shared decision process is very important. Then eosinophils are not driven by the cytokine that is targeted, and you need to switch, or there is inadequate dosing and you have to go, for example, from subcutaneous to intravenous, or there is immunogenicity and then you have to switch, or there are other immune mechanisms like autoimmunity driven by the eosinophil derived products. Then again you have to refer to a specialized center. There are many more research gaps in the and we looked at all of those and we recommended some plans to address this. For example, for long-term safety data, post-marketing surveillance will be extremely important using the severe asthma registries. Or, for example, the correlation between the biological and the clinical response, we need well-randomized RCTs as an example for personalized medicine. Last but not least, because we are going through these pandemics and we were requested to come with a response, and it's also part of the EAACI guidelines, how we deal with the biological. And during the COVID infection, we looked at the immune response occurring to COVID. And you can see here that after the initial innate immune response, which kicks early, you have the adaptive immune response. First, Th1, Th17 inflammation, which is dampened by the type 2 and by the adaptive immune regulatory response. Now, because the recovery phase, the restoration of tissue integrity is very much dependent on type 2 and regulatory adaptive immune response, we thought that it's quite important to maintain the biological treatment if there is no infection, because the patient has to be well controlled. But when you get the COVID infection, even if it is asymptomatic or mild, moderate or severe or critical, it's prudent to withhold the biological treatment for a minimum of 2 weeks until the patient is recovered, in order not to hamper the recovery phase by dampening the T2 immune response. And then reestablish the pre-infection treatment and assess disease for severity and control. With that, I would like to close. All the guidelines are published, and what I mentioned are published in our journals, especially in allergy, which is the leading journal in the allergy field. And I would like to invite you to send the best of your science to these journals. Thank you.
So now I'm ready to take questions into Q&A. If you have any questions, please put them to the chat. We have a couple of minutes to answer your questions. Yes, of course. There's a very important question. patients' reported outcomes, and can we use them in the long-term assessment of efficacy? Of course, this patient-reported outcomes are very, very important, and we should very well use the mobile health technology and artificial intelligence to analyze them. And this was addressed in some papers. I showed here the paper of Jean Bousquet showing that this is a very good tool for the follow-up of the patients. Yes. This patient-reported outcomes is part of the P5, P6 medicine, where the input of the patients is very much considered, of course. So, are there any other questions? I wonder, with your current study method approach, will the sample size be smaller or larger? It will be larger, of course, yes. Sample size effect, yes, it will be for sure larger with this approach, yes. All right, I cannot see any other questions, and we are exactly on time. Thank you very much for your participation, for your questions. I would like to thank all the speakers. I would like to thank APAACI for inviting us, and I wish you a very successful Congress. Thank you very much. Bye-bye.